Thank you. Firstly, thank you very much to the organisers for this opportunity to present this work today. As you said, I'm going to talk about metabolite changes that occur in obesity and after weight loss. So I think everyone's very well aware from the state of the world right now that we're in the middle of this obesity epidemic. In the US, 69% of people are actually overweight and about 34% or so are actually obese. And this increase in body weight in recent years has really been a culmination of two important factors. Firstly, we're now eating more food than we ever have done before. In the last, between 1970 and the year 2000, caloric daily consumption on average has increased by 530 calories. On the other side of the coin, we're actually using a lot less energy than we ever used to before. With decreased energy expenditure, decreased physical activity, this has declined about 32% in a sort of similar kind of time frame. So overall, we've ended up with this very increasing population of increased body weight, which leads to the fact that a lot of people are actively actually trying to lose weight, with the current statistic being about 54% of the population are actively trying to lose weight. So we all know weight loss is hard, but interestingly, what seems to be even harder is maintaining weight loss. So the statistics say that 75 to 95% of people that lose weight will actually regain this weight within 3 years. This is an example here from a popular TV show, The Biggest Loser. This guy entered the show at 407 pounds, underwent a very successful weight loss regimen, losing over 200 pounds, but within a few years after the end of the show was back to pretty much where he started. And this is a very common thing amongst most people that go on a diet. So what could be some of the reasons why people are predisposed to regaining this weight? One of the hypotheses is actually that there's a physiological compensation that happens after weight loss, and actually we have a slightly different metabolic rate in people that have lost weight compared with an equivalent lean individual. This has been shown in both human and in mouse studies. There's also some evidence to show that perhaps hormonal adaptations that occur in the obese state persist beyond weight loss and stay to then predispose these people to actually regain weight due to lasting changes in some of the hormones that are important in controlling appetite. So centrally, food intake is controlled by an area of the brain called the hypothalamus. And the hypothalamus is important in integrating signals for satiety, how full you feel, and the reward value of food. And this can affect neuronal firing of different types of neurons that then encourage us to eat food or to abstain from eating food. And together, these things add together to finally contribute to what our final body weight's going to be. We know that in obese people, we see dysregulation of many different peripheral organs, including the liver, the adipose tissue, and the muscle. And these organs are particularly important in glucose and lipid metabolism in general. These organs also secrete many hormonal and nutritional signals that then feed back into encouraging us to eat food. So we've tried to model some of this to some extent in a mouse model. And this involves, in our model, feeding mice just normal chow in the first part of life, and then dividing them into 3 different groups. One group are fed a standard low-fat diet for the chronic full duration of the study. A second group are fed an equivalent high-fat diet. And the third group, this more interesting group, are mice that we develop an obese state over the course of 9 weeks, and then we switch the diet to a low-fat diet to encourage weight loss, so that at the end of the study, these mice physically and their insulin sensitivity is equivalent to a low-fat diet-fed mouse that's never been obese, but actually these mice have had a long period of their time in life where they've actually been in an obese state. So what we're trying to do is look at changes at both the transcriptional and at the metabolomics level to say, are the changes that occur in these different phases that are induced by obesity and some that are then reversed after weight loss, and would call these reversible changes. These kind of changes in metabolites and genes would be potential targets for insulin sensitization. And then on the lower part of the slide shows genes or metabolite changes that are induced in the obese state that then stay at this new induced level or repressed level. And we think that these then might play some sort of role in driving us towards a weight regain. So we're looking at changes in the transcriptome and the metabolome that may return to the pre-obese state after weight loss. Do they change or do they not change back? And we're looking at lean, obese, and diet switch mice. We've taken a number of different tissues that are particularly relevant to the obese condition, including liver, muscle, adipose tissue, the plasma, and the hypothalamus. So at UCSD, we've conducted RNA sequencing studies on these same tissues, But due to the West Coast Metabolomics pilot grant funding mechanism, we've been able to also extend these studies into two different forms of metabolomic analysis. Firstly, the lipid mediator analysis in collaboration with John's lab, looking at endocannabinoids, oxylipins, and ceramides, and then in collaboration with Oliver's lab, looking at overall lipid metabolism. So just as a very basic and general thing, we're defining reversible and irreversible changes. Reversible changes are things that are induced in the obese state that then go back again after weight loss, and irreversible changes are ones that are induced in the obese state and actually stay at this new high level. And there's a Venn diagram there just showing how we categorize the different changes. So from an RNA sequencing transcriptomics approach, we see that a large number of changes are induced in the obese state as we would expect. These are the ones shown in green. Particularly in the adipose tissue in the liver, we're seeing a very high number of transcriptional changes. We've then categorized these into both the reversible and irreversible changes and show that this paradigm as a whole does hold that there's a large number of changes that are actually induced in this obese state, and after this 9 weeks of diet switch, stay at a particularly different level than they were in a lean individual. So to look at this from a metabolomics perspective, this data shows there are results from our targeted approach looking at the oxylipins, endocannabinoids, and ceramides. We found 32 different lipid mediators that were actually changed in the obese state. The ones in green are actually reversible, so as expected, we thought a large number of these would be reversible after weight loss. So 25 out of 32 of these actually moved completely back to where they would be in the lean state that have never been obese before. But a couple of them are particularly interesting that seem to be completely irreversible. So I'm just going to mention a few of those in particular today. This is the same data but shown in a heat map representation, the low-fat diet on the left, the high-fat diet in the middle, and the switch on the right. Anything that was induced is higher is red, and anything that's low is blue. And the extent of the color kind of represents anything in between. So you can see the green ones labeled green are reversible, and the pink ones are irreversible. I want to pick out just one example for today of one of these irreversible metabolites that we think could be important potentially in this phenomenon of weight regain. So prostaglandin D2 levels were particularly high in the obese mice, and this metabolite stayed very high even after the diet switch. The graph on the right there shows the exact levels of it at the different stages. But we've been able to now look back at our RNA sequencing data in the same extent to say what happens to the synthesis of the enzymes that are involved in the synthesis of prostaglandin D2. Do they also follow this same kind of paradigm? And interestingly, the synthase responsible for the synthesis of prostaglandin D2 in adipose tissue, this hematopoietic form, seems to be highly induced in the obese state and also stays high after the diet switch. So looking like two parts of these evidence point towards the fact that this is indeed induced and stays high in obesity. And just from a brief search of the literature, it was kind of curious to me to see that actually there's a defined role for prostaglandin D2 in food intake, so that people have shown cerebroventricular, so direct injections into the brain of prostaglandin D2 stimulate food intake through the DP1 receptor. So it seems that this has a hyperphagic effect. And our sort of question that we're going to go on and now try and address is, does the peripheral levels of adipose tissue secretion of prostaglandin D2 really play a role in food intake, and could this potentially be something that is permanently changed after the obese state that could predispose people to being more hyperphagic and encouraging weight regain? And so just briefly, our global lipidomic analysis of this same study, we identified a large number of known metabolites that change in the obese state. A large number of these are also, as expected, reversible, but a very large number also are irreversible or indeed not fully reversible. And just as a really sort of general observation, we see that actually there may be a sort of tissue-specific difference to how reversible the changes are in the obese state. So in the liver and the plasma in particular, we see a large number of these reversible metabolites. And just by general observation, the muscle and perhaps the adipose tissue as well also show a trend towards perhaps more irreversible changes. So things that are occurring in the muscle and the adipose tissue are less forgiving and seem to be stay around longer after obesity has normalized back to a lean state. They're still persisting past that time point. And to look at it from a very overall lipid class perspective, many of these things have been very well shown by many other studies of the types of classes of lipids that would expect to see changing, especially in the plasma, in obesity. The starred ones here show the ones that we found are significant changes. Red shows an increase on a high-fat diet, and blue a decrease after the diet switch. So you can see in the plasma and also in the hypothalamus and in the liver, we see things that are increasing in obesity that then decrease again after the diet switch. But generally with the adipose tissue and the muscle, we're seeing changes that are induced in obesity that persist beyond after weight loss. So our main general conclusions so far are that the majority of metabolic changes that are associated with the obese state are actually reversible after weight loss, as we would assume. These track very closely with body weight and insulin sensitivity. And that changes in some metabolites in some tissues appear to be more reversible than others. There seems to be a slight trend towards liver and plasma being more amenable to reversibility, and muscle and adipose showing a sort of longer changes that seem to be less reversible. And there's just one example that we've kind of begun the ideas of how to follow up is looking at this prostaglandin D2 and thinking that perhaps changes in this metabolite may play an important role in weight regain after obesity. So lastly, just to thank the people at UCSD that have helped with a lot of this work, and our wonderful collaboration with UC Davis in both Oliver and John's lab that have enabled us to actually generate this data. So thank you very much.
So questions?
Hi, it was a really interesting study. I was curious, in your switch diet mice, you said that they came back to a normal weight. I was wondering if you've done any further analyses like body composition, DEXA analyses to see if they truly lost all fat mass, lean mass, or whether their liver weights had changed at all.
Yeah, we've done some sort of crude analysis of just measuring the different weights of different depots, and they're fairly similar to the low-fat diet mice. There's a few subtle differences, but generally the low-fat diet mice are sort of always increasing in body weight, so they kind of, by the time you've got that whole 18-week study, they kind of end up roughly in the same place.
Thank you. I have a question regarding the quality of the dietary fat that you used.
Mm-hmm.
So was this a lard base or was it palm oil?
It's a lard-based
Okay.
research diet, 60% high fat.
Have you done any comparison? I mean, how do we separate out diet effects from obesity effects?
Right, so all we have is the low-fat and the high-fat diet are matched for everything except the fat content.
Well,
So
but the low fat
obviously-
is, of course, a high carb.
Right,
Yeah?
so that's all we've been able to do so far in this initial paradigm is to start with that process. We've got some other studies ongoing to look at a different feeding model to generate obesity in different contexts using an intragastric model to try and feed high amounts of carbohydrate to equivalent calories of the high fat to then determine fat-independent effects on obesity.
And bringing the animals back with the weight loss, I missed that. Was this a switch back to a high carb?
Switch back to low-fat diet.
Oh,
yes.
low fat, high.
Mm-hmm.
Yeah. Okay, thank you.
Very nice study. The question that I have is, did you look at the energy expenditure?
We didn't actually measure it in chambers, no. So we've done it in other studies and equivalent studies, but not with these exact mice.